Randomized controlled trials and clinical trials. RCT is a study design that randomly assigns participants into an experimental group or a control group. As the study is conducted, the only expected difference between the control and the experimental group in a randomized controlled trial is the outcome variable being studied. What is RCT? It is prospective, so it's looking forward in time. It is in contrast to retrospective, which looks data that already exists, such as data available in medical charts. RCT is interventional, which means an action is performed or investigators must implement a treatment according to study protocol. It is in contrast to observational, where investigators document the natural course of events. Typical flow of an RCT. First is assessment for eligibility of patients. If patients is ineligible, then they have to be excluded from the study. If the patient is eligible, then the patient will be enrolled and will be informed with all the procedures involved in the protocol. Randomization will be done and a patient will be assigned into an experimental arm and a control arm. The control arm may be inactive placebo or active standard of care. And after a period of time, they will be followed up to a certain outcomes. Why conduct an RCT? RCT is performed to establish cause and effect. High degree of confidence that the results are attributable to the study intervention and not something else, if the trial is well designed. RCT is very useful to establish safety and efficacy of new drugs. It can compare safety and or efficacy of two existing drugs. According to the hierarchy of evidence, where in vitro test tube research is the lowest, followed by animal research, then ideas, editorials, and opinions, case reports, case series, observational studies such as case control and cohort, and then the randomized controlled double-blind studies. So it means that the data that is gathered from RCT is considered as a reliable evidence for clinical care. General strengths and weaknesses of RCT. The strengths, it can define study population. It also has compliance maximized through study protocols, and it can establish cause and effect. Some weaknesses includes the exclusion of many patients who may need treatment. Another is short duration and modest number of patients limit ability to detect rare or long-term adverse effects. What are the different RCT design types? We have parallel arm, factorial, and crossover. For parallel arm trial, this is the most common type of design. It involves two or more groups, which are the comparison control group, and one or more experimental treatment groups. It randomly allocates patients to exposure. In parallel arm, subjects remain in their group for the duration of the study. It is good for assessing treatments that have long-lasting effects and can prevent or cure disease. Parallel arm trial is done with randomization. After randomization is done, Control, treatment arm 1 and treatment arm 2 will be performed simultaneously. Parallel arm trial is very good to identify cause and effect when interventions are mutually exclusive, but it also requires more patience than other designs. Different patients also in different groups can result in between group variation. The second type of RCT is the factorial trial. In this type of RCT, there is more than two active therapies and their combination is given to the patient and being compared to the control group. Therapies should have common goal of achieving same outcome. Patients assigned to one of the following for the duration of the trial: the treatment 1, the treatment 2, the combination of treatment 1 and treatment 2, and the control. This is an example of factorial trial using ramipril and rosiglitazone. Factorial trials allow for multiple hypotheses to be assessed. From the example on the previous slide, it can help answer the following question. Is rosiglitazone effective? Is ramipril effective? And is the combination more effective than ramipril alone? Is the combination more effective than rosiglitazone alone? So the strengths and weaknesses for factorial trial are the following. For the strengths, it is efficient for testing multiple hypotheses. Less patients needed to assess multiple intervention, and it would need multiple parallel arm trial to test the same number of hypotheses. It can detect synergistic or antagonistic interactions. But the weaknesses are different patients in different groups can result in between group variation. Also, it has higher risk of making an incorrect conclusion. Therefore, possibility of anticipated interaction between two treatments may occur. The next type of RCT is the crossover trial. Patients participate in each arm of the study in random order. It is appropriate to use if outcome is reversible and study is short-term, and also if disease state is stable and treatment is symptomatic. But this is not appropriate for curative effects. The process of crossover trial is presented in the diagram. After randomization, the first group for control and the first group for active treatment is assigned and period 1 will begin. Afterwards, a washout period must be obtained before proceeding to period 2, where the original control group will participate this time in the active treatment, and the original active treatment group will become the control group. This is a specific example that compares tramadol controlled release and tramadol immediate release in the treatment of chronic non-cancer pain. Strengths and weaknesses of crossover trial. For the strength, all patients receive all treatments. So each patient serves as own control and no between group variation, which is advantageous compared to the first two types of RCT. Also, it requires fewer patients than parallel arm trial. But the weaknesses are it has longer duration, potential for carryover effects, time or an order could be impacting results, and it requires more complex statistical analysis.